So I'm very pleased that Jerry, realising that there was an imbalance of points over counterpoints, actually spent most of his discussion on counterpointing. So I'm not going to repeat everything he said, but basically try to introduce to you other ways of thinking about this issue. So I just want to say I have conflicts of interest. I started 2 new virtual companies this year just because I have nothing better to do, and I consult with virtually every company in North America about antimicrobial discovery. So Jerry's already really mentioned this. The antibiotic development paradigm as it's been practiced for years is fractured. And there's no question that antibiotics underpin, they're not just important for treating infections, but they underpin essentially all of modern medicine. And we really have a very dry pipeline. We have some compounds in the pipeline, but really I would say almost none of these things are game changers. And contrary to popular opinion, this is not because we haven't done any research. Large pharma spent massive amounts, billions of dollars, on trying to develop new antibiotics. But for 30 years, really nothing that would make a big difference. In terms of veterinary antibiotics, the FDA has clearly stated, according to companies that I consult with in the US, that if a compound comes up and is developed for agriculture and has potential human use, or even a relative of that antibiotic has potential human use, that it will not be licensed for agricultural use. So these two areas in some way are quite distinct. So there's no question that there's an urgent need for different ways of treating infections. So the conversation worldwide has been about antimicrobial resistance, but it's really the tip of an iceberg. We have to start to consider when do antibiotics not work? And believe me, the problem is much bigger than 700,000 people dying a year. So resistance has been defined in a lot of reports by acquired resistance, mutations, plasmas, genetic elements, etc. But there's a syndrome called sepsis. It has a 30% death rate. The estimate of the number of deaths per year are 5 to 17 million. And for all of those in the room who have grey hair like me, you're in the sepsis era, by the way. You're starting to get into that danger zone. So it has a massive death rate. A massive number of people die, between 8 and 16% of all the people who die on the planet. And the frontline treatment is antibiotics. So people die because antibiotics fail. Chronic infections are 2/3 of all infections, according to the US CDC, and they're multi-drug resistant, but not because of mutations, but because of the growth state of growing in biofilms makes them resistant. So here's a huge area. There's never been a single compound developed for treating biofilm infections. And individuals who have disturbed immune systems, which is increasingly common in this era, people under chemotherapy with immunosuppressive disease, genetic diseases, and massive injuries and burns, these people also have infections that are extremely difficult to treat because they don't get a boost from the host immune system. So these things are rarely taken into consideration while counting how many people are at threat from the loss of antibiotics in our society, and also when thinking about solutions. So a bunch of us came together in London about a couple of years ago now, and we started to think about alternatives to antibiotics. And we've also, I consider these to be adjuncts or adjuvants, like Jerry mentioned. They're probably going to be used together with antibiotics. So the approaches include antibodies, vaccines, probiotics, lysins, wild-typed and engineered phages, immune stimulation, antimicrobial peptides, innate defense regulators, and antibiofilm peptides. And I'm going to try and cover all of these in about 7 slides. So sepsis, I mentioned, a huge issue. It happened to be World Sepsis Day yesterday. So we've basically been really interested in this situation, and we found a gene expression signature that seemed to associate with sepsis when patients were still in the emergency ward. We proved that it was present in all of the data sets that are out there in the literature, with a staggering p-value with lots of zeros and then a 1. The accuracy of this signature in diagnosing sepsis through this immune cell reprogramming signature, or a kind of a cellular amnesia signature where the cells forget how to respond to a bacterium, is highly accurate. We then went and did a clinical study, and we showed that sepsis predicts severe sepsis while patients are still in the emergency ward. And we also showed that it predicts organ failure. And just yesterday at Sepsis Day, we announced that we're going to start a 1,000-patient clinical study to try to firm this thing up in a large multi-centre study. So this is a different way of thinking about diagnostics, where we're diagnosing the patient condition, which is aberrant and leads to the failure of antibiotics. But we can also think about this in terms of a new type of therapy, where instead of directing our therapy to the bacteria, we direct our therapy to the underlying defective condition in the host. So we try to reverse the cellular amnesia condition. Vaccines are the single most cost-effective intervention in medicine. They're used on a population basis and they're usually quite inexpensive. So here's a project that I'm involved in called REVAMP, which is funded through Genome Canada. It uses an approach called reverse vaccinology. We take genomes, we bioinformatically mine them so we identify all components that could be antigens. Then instead of kind of picking and choosing one or two, we just clone, actually we synthesize the genes for all of these antigens. So we've synthesized several hundred in the last year for 2 different organisms. We then express these and test them in actual cattle models. And we're fairly advanced in this towards moving towards a new vaccine. And this type of approach, really high throughput, 1.5 years to get through to the stage where we're getting close to vaccine testing. This is the sort of approach of the future in vaccines. Immune modulation is another strategy that's already used in both the animal world and the human health world. It's much more highly utilised in antiviral and anti-cancer therapy. And it's based on the concept that successful therapy against infections requires help from the host in terms of an immune response. So we've documented a whole bunch of cases where, in fact, there is immune modulation therapy that is useful. Immune modulation is a really nice approach for One Health because it doesn't give rise to resistance because you're not changing the bacterium, you're changing the way the host responds to the bacterium. And one example is we've taken this single peptide here, and excuse what happened here, but we've demonstrated it's effective against Staph aureus infections, multi-drug resistant tuberculosis infections, cerebral malaria infections, in wound healing models against all of these things, including viral infections, and recently we showed it was effective against Candida infections, also sterile inflammation and malaria. So we've got an incredibly broad range of activities that we can use as an adjuvant or adjunct to normal antibiotic therapy. We've also been really interested in the biofilm area, and we have managed to show that some of the peptides we work on also have very, very broad spectrum activity. Essentially, we've demonstrated this against all of the escape pathogens, which are the most resistant in our society. They show tremendous synergy with conventional antibiotics. So this is activity in the same NDM strain that Jerry just mentioned, a carbapenemase, plasmid-encoded carbapenemase producing Klebsiella. And we can show that a tiny amount of meropenem that is usually to which this organism is totally resistant, together with a bit of peptide, causes activity. The peptides work on a bacterial stress response, and we've demonstrated good activity in animal models, especially in abscesses that are virtually untreatable by antibiotics. I felt I should mention probiotics, although Tom Louis is going to talk about this in much more detail in a few minutes. But probiotics basically do have some Canadian roots, so this is a Quebec product. It's the use of bacteria in preventing infections. There are many theories as to how they work, and one of them is exclusion, another is immune modulation, which we kind of favour because we're one of the proponents of this. This is already used successfully. So probiotics, or helpful bacteria, are used as over-the-counter medicines for strep cell in the form of strep salivarius, and it's been clinically proven in controlled clinical trials to be effective against strep throat, halitosis and chronic sore throat in children by this little New Zealand company. It's also been shown, and Tom's going to talk about this in greater detail, that fecal transplantation is effective against the Clostridium difficile. And the thing I like the most is this phenomenal study done in 10,000 premature, very low birth weight babies to reduce sepsis by 40% and enterocolitis by 40% in the most susceptible populations of infants. And these results have now been reproduced in a similar size study in India. So there's no question this is a massively pennies a day dose costing effective therapy. And now we're increasingly becoming knowledgeable of the fact that consortia of defined bacteria can replace the normal microflora. And then finally, I'd like to talk about phages and lysins. The story's not quite as good here, but the idea is that phages basically cause bacteria to explode. And one can use consortia of natural phages, as has been used in a place in Russia called Tbilisi. One can use phages that are engineered to utilize common receptors on bacteria, so one phage will hit lots of different types of bacteria. Phages that produce Cas9 CRISPR, which is kind of a bacterial genetic engineering tool, or toxins that can knock out the bacteria, or the proteins from the tip of these phages called lysins that are enzymes from phages that destroy pathogens. And there's a US company that just entered phase 3 clinical trials, which I also consult with. So phages self-propagate as long as their bacterial host survives, so they're a very attractive kind of, you can add them in low doses and expect the bacteria themselves to propagate their own means of death. Several trials are underway, but there have been some recent very prominent failures, including a very large study that was conducted in India earlier this year, or it was reported earlier this year. More research is needed to understand phage biology, to create new phages with broader host ranges, and to detect and engineer lysins. But this is an extremely promising mechanism going forward. So in conclusion for what I've said, I've tried to show you some stuff that is different from antibiotics, but achieves the same objective of knocking down bacteria. I believe this is something that we could come up with as a made-in-Canada approach of using compounds that by themselves perhaps have some activity and could be used in the agricultural field, or they could be used adjunctively together with antibiotics. So there's no question that resistance is... Sorry, there's no question that I did the wrong thing there. So there's no question that resistance is very complex. But I would say that we really need to start considering all of the situations when antibiotic therapy doesn't work. There's no problem. 10 million people, 2050, there's probably 10 million people dying of antibiotic failure right now. So the problem is not in 2050, the problem is today. Given our recent dismal success, we have to ask the question, what if we never discover a new game-changing antibiotic? And I would like to say that alternatives to antibiotics provide new hope. And nobody's shown a picture of people who actually do work, but these are the guys in my lab who did all the stuff I mentioned. Thanks.